So just share here a little bit schedule of this course. I will start by introducing, why is the Cellosaurus, why did it exist, and what are the other resources that exist on cell line in general, and so basically an introduction of about 20 minutes. Then we'll have a big part, maybe a little bit heavy, which is a detailed description of the content of Cellosaurus. That would be the beef of this course. Then we'll do a 20-minute pause because you'll be exhausted after that. And then I will look at use case of the Cellosaurus for about 30 minutes, then go to future developments and conclusion, and then we reserve the 50 minutes for question and answers. But as Patricia said, you can interrupt with question, or if you feel shy to say them loudly, you can type them in the chat and Patricia will forward them to me. Okay, so let's start just with one slide about cell line. They're used by labs in academia and industry all over the world. Probably don't need to tell you that. And we'll see this in more detail. Some are acquired from cell collection and others are transferred between labs. And this we will go back to it because this is quite important for a number of reasons. One which has to do with the information on the cell lines, other about cell line integrity. But we'll cover this in a lot of details. And of course, there is a numbers of variety of cell line. People know about cancer cell line like HeLa. There is, of course, embryonic stem cell, iPSCs, hybridomas, and so on. So there's a whole menagerie of different type of categories of cell line. And in terms of resource, there are a lot of different resources around cell line. We'll see them in this introduction. So ontology and many specialized databases. But until Cellosaurus was established, there was no single resource where all this information was available. Now, you could think that the Cellosaurus then was created with a rational idea of creating such a resource, but it didn't really happen like this, in fact. It started, during the early development of NextProt, which Patricia alluded to a few minutes ago, so which is a knowledge base on human protein. And in this knowledge base, we wanted to annotate as precisely as possible the biological entities which are used to perform experiments on human protein. So of course we needed species, but this we could get, through the NCBI taxonomy, database. We needed information about tissue and organs. There's different ontology on tissue and organ, and of course cell line. So naively, we thought that we could use existing control vocabulary or ontology to annotate the cell lines. And so we looked at, and we couldn't find anything which we could use because there were resources, but none of them contained all the cell line we needed. And at that time, it was not a lot. We needed only a few hundreds. We were annotating only a few hundreds. Needed information about, I think, less than 200 cell lines. But even so, that this was a very small number compared to what the world of cell line is, and you will see this later. We could not find a resource which had all of the cell line we wanted to point to. So the idea was to create a small cell line thesaurus. Very simple, just cell line names and synonyms and maybe a few references. And thus the name of the Cellosaurus, cell line thesaurus. So that was the plan. Just nothing very fancy, just, you could say just a dictionary, a list of cell line, and that's it. But of course, nothing that you plan goes the way you want, really. And more people became interested in this small thesaurus. And they asked us if we could add things, more cell line, of course, but also broaden its scope in terms of information relevant to the described cell line. So basically, what was a small thesaurus in inception about 9 years ago evolved into a full-fledged knowledge resource. So you can see this by looking at this slide. You see here one entry inside the Cellosaurus. It's a mouse cell line, a macrophage mouse cell line, as it was in release 1, and what the information is in release 37 for the same cell line. So you see that, there is, you don't need to look at the details of this entry, but visually you can see there is much more information, and there is also a variety of information, which has increased, between this release 1, which was in 2012, and release 37, which was last month. Now, I mean, one of the things we wanted to use, as I was telling you, was ontology of cell lines. And what exists in terms of ontology, the world of ontology and its correspondence to cell line is quite, interesting. There is a cell line specific ontology, which is called cell line ontology, CLO. So that's the first thing where we wanted to use when we were annotating an XProt. But the thing was that there was a lot of terms in it, 39,000 terms. 30,000 describe cell line, but with a high level of redundancy because there can be many entries for a given cell line as there is distinct providers. But it's high quality in defining cell type and cell categories. Unfortunately, as I said, 30,000 cell terms seems a lot, but it's a lot less cell line because of this redundancy. And even 15 or 20,000 different cell line, which is probably what it contains, is not the whole world of cell line, as you will see. And of course, this is an ontology which describes in an ontology way what are the cell type and cell categories, but it's not a knowledge base on those cell line. It doesn't describe in details of cell lines. Since there was an effort made by Molecular Connection in India to create a cell line ontology, and unfortunately, they stopped maintaining it, so there was never more than 500 cell line into it. And then there are efforts by different ontology to cater with cell lines. The best, I would say, comprehensive one is the Brenda tissue ontology, which has inside this tissue ontology, which covers both tissue, organism, everything which is needed to annotate Brenda, which is an enzyme database. There are 2,360 cell lines described. It used to have duplication, erroneous entry, but we have a collaboration now in the last 3 or 4 years. And every time there's a new release of Brenda, we look to it, we can tell them if there is duplication and so on. So those 2,360 entries in Brenda really represent 2,360 different cell lines. Then at EBI, you have EFO, experimental factor ontology, which is used to cover the cell line used in context of some EBI database or ENCODE. So it's not covering a lot of cell line, 1,300, and it's quite error prone. There are a lot of duplicates and so on. The idea of EFO is really to help annotate, some of the data resource in EBI. Then there are 2 small, other resources, a beta cell line ontology, which is not maintained, which was only for diabetes research. And of course, the oldest ontology in the life science, MESH, but MESH only describes 25 cell lines. And basically, it's just anecdotal, the fact that there are cell lines into MESH. So as you see, the ontology world regarding cell line is not covering far from everything we need, to represent all of the different cell lines that exist, and also is restricted because those are ontology and not knowledge resource. Now, what is there else? Well, first, before going into real database or bioinformatic resource, one thing which is interesting to look at is how are cell line distributed, because this will allow you to see what is the problem and where the information can be found. Well, some of the places where you can find cell line are what are called cell collection. Those are institutions that distribute from a few hundred up to 40,000 for cordial different cell line. They generally also distribute microorganisms, bacteria or virus, and other biological samples. And most of them are non-specialized. They will distribute all kinds of cell line from a variety of organisms. Some are specialized, and I've put here a few examples, either in terms of which cell lines they distribute or in terms of what organisms they cater for. And some will cater to local users. They will ship only inside a specific country, while other will ship cell line worldwide. Here, and I'm not going to read it, you have a list of the main cell collection, and you immediately recognize the first one by alphabetical order, but which is also one of the most well-known one. It's ATCC, in USA, which distribute both cell line, microbes, virus, and also DNA samples and so on. So those collections are quite important in the ecosystem of cell line. And because they provide not only cell line, but information on the cell line, they're also important in this respect. And we'll come back to that in a few minutes. Now, you also have companies which either specialize in the establishment of engineered cell line, like knockout cell line, or which distributes a number of cell line. And there's also those big laboratory science company like Millipore, Thermo Fisher, and so on. And a lot of those companies so distribute cell line. It can be a few, or it can be many, depending on the companies. And here is a list. It's not, I mean, a complete list, but it shows you a little bit the players inside this world of companies and cell line. One thing which I should mention here, and I won't come back to it, is that there is many companies that exist which distribute cell line, which are called, I call rogue companies. Some people call them pirate company. They're basically distributing cell line illegally. They have acquired them either by buying them from the collection which have the right to distribute them, or from academics. And then, of course, they're growing them and redistributing to everyone which wants it at a lower price than the cell collection, but without doing any QC. And this is really a big problem because there is more and more of those companies. And I mean, unfortunately, they proliferate and they're based in countries which do not really, I'm not going to cite those countries, but they're countries which are well known because they don't really follow, I mean, copyright, I mean, laws, and they basically accept to have companies which are pirating, I mean, biological, in this case, products, cell line. Now, one of the main way cell lines are also distributed is by academic labs. Academic labs establish cell line, and then they will distribute them. Some will do this through their TTO, their Technology Transfer Office, or their biomedical resource facility. But most do that in an ad hoc manner, meaning that a colleague will ask them for a cell line, they will give it to that colleague. And of course, that colleague will redistribute this cell line to another one, which themselves will distribute it, which is a recipe for disaster. Because in fact, as soon as you have people redistributing something and then another person redistributing and so on, knowing what exactly is being distributed can be difficult in some case. And I mean, and I know it firsthand by working in the lab also and having a wet lab to characterize protein, having just, I mean, a small vial with the name of a cell line is not going really to be very helpful a few years later when the postdoc has left to know what was in this vial. And if you start redistributing those badly, I mean, defined entities, you're going to, I mean, cause problems to those which use it. Now, the cell line collection, you could say that could be a good way to get information on cell line. And yes, it is. There are 50 major entities which are distributing cell line, but they're not at all savvy in terms of good bioinformatic practice. It's really, I mean, a nightmare. I mean, we once drafted 5 minimal requirements that those cell line collections should, I mean, follow. So that they should have an individual page for each cell line, so that if you go to them and look for a cell line, you should have 1 page of information at least for each cell line. That the URL of those pages should be based on their catalog number and not just by a name. They create just page based on whatever the person which created the page likes to call it. There should be a mechanism to be aware of new cell line and of those which are discontinued, which are no longer distributed. And this, I'm not going to explain what it is now because there will be quite, I mean, a number of slides on STR profiles. They should make the STR profile available, meaning some information to allow identification of those cell lines. Now, here visually, you see an extract of a spreadsheet, and you don't need to read. I mean, I took away anyways the names of the cell collection, but you see those 5 columns for the 5 requirements. And basically, none of the 50 or so collection are compliant with all 5 rules, and 6 have even 0 compliance. So basically, they don't even have individual page, they don't have a mechanism. Well, you see the rules and you understand what 0 means. Some are changing, but it's taking a lot of time because those cell lines were not thinking of themselves as provider of information. So they're thinking of themselves as provider of biological entities, cell lines or microorganisms, but to transition toward the bioinformatics is something quite difficult for them. They don't understand good reproducibility practice in terms of not of biology, but in terms of bioinformatics. They don't understand that catalog number should be constant and that you should not reuse a catalog number if you change a product and so on. So that's a major issue. I mean, because there is a treasure trove of information from those cell collection, but it's difficult to access. And of course, their websites are generally, I mean, just page of information, HTML with no structure whatsoever. Now, let's go to what exists in terms of real database for cell line. There is, to my knowledge, and I've looked for a long, long time, only one universal database like Cellosaurus. It's the cell line database, CLDB, which is, I mean, developed in Genova, in Italy, I mean, under the auspice of Barbara Parodi. And it was really a great effort. I mean, it's high quality, and it was meant to be a universal, I mean, database catering for all different type of cell line. But unfortunately, it hasn't been updated since 2017, which is not a long time, but still it's 4 years, but it's also only describing 6,640 entries, so about 5,400 different cell lines. So the structure of this database is quite nice. It caters for a lot of information on cell line, but it's basically, an effort which was too small, and which has not been kept in. I think that it's no longer going to be updated anymore, as there is nobody really working on it full time. So now there are specialized resources, and those are quite important. I mean, one of the, I would say the most active and ongoing resource is the Human Pluripotent Stem Cell Registry, which is specific for human embryonic stem cell and pluripotent stem cells. It's very high quality. It's, quite well done in terms of bioinformatics, in terms of, all the tools available through it. You have APIs to query it. It's really, a star example of what should be done correctly in terms of catering for, cell line. Of course, it's specific to one category of cell line, so it doesn't cater for every type of cell line. So it's human only and those stem cells line. But in the world of stem cell line, it's really a star resource. And we basically collaborate quite, nicely with HPS Reg. Basically almost like daily exchange of mails and information between the two different resources.
Amos, there is a question in the chat.
Yes?
There's a question in the chat about the minimum requirements. What do you mean by a mechanism to be aware of new cell lines?
Well, at minima, at least have a page which, describe what's new in your resource, in your, cell collection. Of course, it would be nice to have a type of programmatic way where you could queries and just, send a request and it would give you the last cell line established in that cell collection since the date that you would, but that would be too much to ask them. Just a page telling what cell line were established, recently or distributed recently would be enough. Some of them do that. If I just go back very quickly to the graph, I think that this is the second individuals. It's maybe this page, this third line here, and you see at least 7 or 8 resources of page which describe new cell line, ATCCs, one, DSMZ, and so on. So some do, but not all of them. Okay. Oops, sorry, went too far. So here was at those different type of resource. There's a resource for, also cell line which are used in a framework of the MHC complex to type MHC complex. It's the International Immunogenetics Information System, which has a section on cell line. And unfortunately, there are many dead resources. Here are at least 4 of them. One which was specific on prostate cancer cell line, one which was for fish cell line, one from stem cell, one for insect cell line. All those resources are no longer updated. Not only they are not updated, but they disappeared completely from the web, or they're completely, unresponsive. So then you go to something else where you can find information on cell line. And this is really where you can find a lot of information. Those are also experimental portals and repository, like the Cancer Cell Line Encyclopedia, the cancer dependency map, the genomic of drug sensitivity of cancers, a lot of things for cancer, because of course, people are interested in research on cancer. So you see all those resources are cancer-based. I'm not going to describe them in general. It's basically a huge number of resources which are doing a lot of omics on a number of cell lines. I mean, it can be exome sequencing, it can be a lot of different things, but a lot of omics research or drug response research on a huge number of cell line, of cancer cell lines, generally between 1 or 2,000 cell lines. And there are also resources for iPSC. And then you have portals which try to integrate all of this data. And most of them are, again, about cancer cell lines. Here you see just a list. I'm not going to go in detail. Those are really great resources, but they basically specialize on one type of category. Most of them are cancer, and they integrate data from those different experimental resources like I showed you before, CCLR or DepMap and others. Finally, and this, but not the least, there is a lot of repository and database which are not cell line specific, but which contain a lot of information to cell line. Things like ARA-Express and Geo, biosamples, Campbell for drug-like bioactive compounds, Cosmic for somatic mutation, the Encyclopedia of Genetic Element, ENCODE, the FCS-free database, which has information on medium which are free of animal products, fetal, bovine free, medium and cell lines. So YARC TP53 database, which has information on all type of P53 mutation, metabolites for metabolomics, PRIDE for proteomics, and so on. I'm not describing all of them. I only put here, a few of them, knowing that there are probably hundreds of those resources, and some have only information on a few cell lines, and some have information on many samples coming from cell line. So the cell line, database landscape is very fragmented. It's really heterogeneous. It goes from things which are just page of information in cell line collection to, defined ontology, but whose scope is not enough to cover everything, to very specialized resource in a lot of different fields. So that leads to Cellosaurus. I'm just putting one slide here which summarizes Cellosaurus before going, of course, in more detail. The Cellosaurus has no information on 126,000 cell lines. So you see the difference between the different resources we saw, which had between a few thousand to a maximum of 30,000 cell line entries. Here you have something which is already 4 times bigger than the biggest second resource. And its scope is everything which is immortalized, naturally immortal, finite cell line, but not primary cells, and invertebrate, but not plants. So all of plant cell line is very specific to people working on plants. A lot of different information items. You will have a lot to see in a few when I start part on the description of the content, a lot of reference and cross-reference. As you will see, it's available on Expazi, downloadable in different type of format. We'll go into this in quite a lot of details. And it's part of a number of initiatives, and it collaborates with a number of databases, including, as I was saying, because this, collaboration is quite nice with the Human Pluripotent Stem Cell Registry. And since about a month ago, it's been recognized as one of the Elixir core data resource. Now, if I look at the timeline of Cellosaurus before going into the second part, so as I told you, it started as a control vocabulary for Nextprot, and it was released with the name Cellosaurus in the beginning of 2012 with 6,800 entries and cross-reference to 21 resources and only 8 information fields. And then 3 years later, it became available on the Expazi server. So that's where people could really start browsing to it and go to Expazi to look at what there was in it. And that was with release 12, and people could access it by FTP. And then it became also the cell line resource for what is called error IDs, which in the context of the Resource Identification Initiative. Don't worry, I will explain what this is a little bit later. And then we introduced STR profile. Again, I'm going to explain what those are 5 years ago. It became a member of the International Cell Line Authentication Committee. This also will be discussed when we discuss STR profile, because all of this has to do with integrity of cell lines. It was first distributed in XML in 2016. The paper describing the Cellosaurus was published in 2018, and we published a tool to look for similarity to, and which we will look also during this course in 2019. And as I said, in 2021, it became an Elixir core resource. And now, as I said, 126,000 entry, 93 resource instead of 21 resource in terms of cross-reference. And we went from 1,000 literature reference to about 21,000, and from 8 information field to 46. Okay, curation strategy. How do we get information in Cellosaurus? This will be 2 slides. So there is 4 parts to get data into Cellosaurus: extracting data from cell line collection product page. And as I already told you, this is not easily automated because all of those pages are quite heterogeneous in nature, and they're basically often just web page, HTML with information. So yes, you can scrap this data, but then you have to manually go through it one by one to try to find information. Now, the second thing is to extract data from external bioinformatic resource that cater for specific data. And this, yes, this can be done with some of the cell line resource or the, different repository, which I described, a few minutes ago. But then the biggest input in terms of data comes from manual curation for publication, about 21,000 papers so far. And still small, but growing quite nicely is submission and personal communication. And this is increasing quite nicely. I'm happy about it. Compared to what I saw during the 20 years or more that I was working with Swissprot, where it was difficult to get people to submit information, it's less for cell line because people are generally quite pleased they've created a cell line and they would like their cell line to be represented into the Cellosaurus. And of course, it helps if they tell me about it and they send information. So there is an incentive for people to advertise their own cell line. So basically people are now submitting information. Now, in terms of how does it work in terms of manual curation? It's like any type of manual curation. You first have to find the relevant papers. And of course, there are a lot of papers cited in cell line distributors documentation, but of course, the easiest way to find paper is to do PubMed and Google search. And for new paper, to have Google Scholar alerts for new publication. Then you retrieve the full text. And using a variety of approach, we obtain more than 99.9% of all the papers cited in Cellosaurus. So either we had access to it through a library, information systems, or we used a site, which I won't name here, but a lot of you know about it, which allows to get papers, full text of paper, or, and that was the third source, we went through our university interlibrary system where you can order articles and get them. And we store all those articles locally so as to be able to go back to find missed or incorrectly abstract information. That's quite important. Of course, then you need to read the relevant part of the paper to extract the necessary information, which doesn't mean reading the whole paper, because if it's a paper describing, a biological experiment and only a few lines speak about the establishment or the characterization of a cell line, since the only part which will be read is the part which is relevant to the cell line. And in some case, for recent paper, it means contacting authors to clarify some points or to ask for additional information. Here, the response rate is about 50%. Not 50% after one email, but 50% after 3 emails. One first email, another email 1 month later, and a third email 3 months later, which politely says, well, I guess you forgot about it or you lost this email. And funnily enough, you get either a response with the first one or the third one. The second one, I don't know why, doesn't serve anything. When you send an email 1 month later, you don't get a lot of more response. But the one 3 months later seems to be like a reminder to people, oh yes, I should have answered that email and feel bad about it. I mean, that's maybe a sociological conclusion, which is not, I mean, very serious, but that's what I observed during the last few years. Now, let's go to this detailed description of Cellosaurus, going by different, I mean, subtopics. Let's start with the fact that a cell line needs a name. Now, as you will see, there is a big naming, there are big naming issue about cell lines. So we will talk about the name, we'll talk about synonyms, misspellings, accessions, and we'll speak about this thing I just mentioned before, the resource identification initiatives. And so let's start with the naming issues. So the first problem is short cell line names are a disaster. And here are some examples, 10 different names of real cell lines, which covers 37 different cell lines. So the cell line PC3, which is a well-known prostate cancer cell line, happens to be also a pancreatic cancer cell line, and also another cell line in another species, which has nothing to do with it. TK is the name of 5 different cell lines, which have nothing to do with each other. So it's a recipe for disaster to have a short names. So some people create longer names, which is good, but some people will in fact abbreviate them. Here is the name of a cell line, FG2/C3A. And of course, people being a little bit, I mean, sloppy will just call it, or lazy will just call it C3A and forget about the FG2 in front of it. And of course, there is more than one C3A cell line. There is no nomenclature rules which have been proposed, I mean, except two. One which was proposed in 1970s for insect cell lines, which is quite well followed, and one recently, 2018, for human ESCs and iPSCs. But I mean, so ICLAC, and I will explain after what is ICLAC, is trying to now go in a direction of having nomenclature rules, but we know what it is. A nomenclature rule will not work if there's nobody enforcing it. It was true for gene name. I mean, nobody was following official gene name until paper told authors you have to follow the Human Genetic Nomenclature Committee guideline or the Flybase database and so on. So until journals tried to enforce things, nobody would follow rules. And there is a huge number of misspellings. We'll go back to it. Okay, so name. So the name of the cell line recorded in Cellosaurus is the one most frequently the one used assigned by its creator. If somebody creates a cell line, well, they should be able to name it. Except when it is problematic, like we said before, too short or identical to an existing one, and that another one has been assigned by a subsequent publication or cell line collection. Sometimes we don't have a choice. If it's short and nobody has given another name, then we have to do something about it, and I will show you in a few seconds what we do when two cell lines have the same name. Casing can be important for cell line. Here are two cell lines, Bob and Bob. What's the difference? There are different lines, cell lines. Now, as I said, the name has to be unique in the context of Cellosaurus. So when two or more cell lines share the same name and we cannot change it because nobody has published another name for one of those cell lines, we append to those names a short description of the cell lines. So here you have two cell lines called AML14. One is a human leukemia cell line, and the other one is a mouse liver cell line. So when you go to Cellosaurus and you type AML14, you get two results, but you see that one is for this human leukemia and the other one for the mouse liver. So that explains something you may have seen in Cellosaurus, and you may have wondered why you have seen those square brackets. Okay, then synonyms. Well, we try to capture as many as possible. And as for name, casing and punctuation are important. And we also capture not only abbreviation, but expansion of the name. I need to show you examples for this. Here, you have two famous cell line, He line, MCF7. And what I say by abbreviation, I mean, is easy to understand, but what I mean by expansion would be something like this. MCF7 stand for Michigan Cancer Foundation 7. So nobody generally use that name in a paper, but like this, it allows people to know why that name was given. Or here, SUDHL4 is Stanford University diffuse histiocytic cytic lymphoma 4. Is there a question? Because I saw a lot of...
Yes, there is a question. Perhaps we will cover this later, but what would you consider to be a minimum data set required to describe a cell line, and how can authors be encouraged to record their cell line data in a systematic, bioinformatic friendly manner using metadata and ontologies?
Okay, I'm going to describe this at the end when I speak about future developments, because what we would like to develop is a tool to develop so that people can enter themselves information on cell lines. And this tool will be complex because it all depends on the type of cell line people are entering. What you enter as information for, I mean, a cancer cell line is totally different than what you enter for information for a genetic disease cell line or for a transformed cell line in one field or another. But I go back to that, okay? Because that's described at the end of the course.
Thank you.
Misspellings. I mean, we record misspellings. Here you have two occasional misspelling of the EC293 cell line. And also if it is specific to a publication or database entry. The first one here is a misspelling. The first one, EC293, are misspellings that if you look in Google, you find a lot of different papers which have this misspelling. So we don't record the list of all those papers. But once it's specific to a paper or to a database, we just, I mean, send enter this information. So you could say that's not nice. You're not nice to those database in paper because you show that they made this mistake, but we also do mistake and we admit them. So here we said that this cell line, which, well, I have to find which one is the real name now, was misspelled EFOP 111 in Cellosaurus between release 9 to 24. So we do also create misspelling. I mean, we try to minimize them, but like everyone, we make mistakes. But then we keep track of those mistakes in terms of misspellings because they could be important if somebody has used one of those releases of the Cellosaurus and thought that the cell line was named like this. Now, you need not only an entry name, a cell line name, which can change over time, but like every resource in the world of life science or every other world, you need a unique stable identifier, the accession string or accession number. Here is the format of the accession numbers or accession string in the Cellosaurus. So there is CVCL, which date back from the time that it was a control vocabulary for cell line. So that's why CVCL, people sometimes ask why. And then a four characters alphanumerical designator. And it can happen very rarely that two or more entries are merged. And because we thought there were separate cell line and we find out there's the same. And one is kept as primary accession and the other is, and there could be more than one, are stored as secondary accession. So here you have the accession numbers of this cell line and two secondary accession numbers. It's the same as with sequence database. And when an entry is completely deleted from database, it's quite rare. It's happened only 36 times so far. It is recorded in a file available from the FTP site. And of course, when you search in Cellosaurus with those accession, it will tell you that this has been deleted and you can see why it has been deleted. Now we go to an important, it's still about names, research resource initiative. They introduced already a number of years ago the concept of what is called RRIDs, Research Resource Identifiers, which are unique and persistent identifier for referencing in literature all kinds of research resource, antibodies, organisms, software tools, and of course cell lines. So this is an initiative which is trying to get journals to force authors to, once they write a paper, to cite those error IDs so that people can go back to different resources and see what exactly has been used in a research paper. So the Cellosaurus is cell line resource for this initiative, and the error ID for cell line are the Cellosaurus accessions. And when you in a paper you want to look at an error ID, it should be prefixed with the word error ID, followed then by the different identifier of the different resource. And you can, of course, search error IDs. There is a portal for it, which is mentioned here. And you go to it and you can search for error IDs for all those different kind of resource, not only cell line, but antibodies, as I said, strain of bacteria, organism, and tools, software tools. Here is an example of a paper where the experimental section correctly, mentions the different cell lines that they use. They do give the name of the cell lines. They say where they obtained it, like ATCC, or from from here another company, CH3 Biosystem. But then they do put the error ID of the cell line, which allows people to go back to the database and see what they did, what they used, sorry. Okay, last slide about naming. There is more and more papers which use those error IDs. And now, so after 4 years since we joined, this initiative, there's already 4,200 papers which have error IDs, mentioning cell lines. Okay, now that we know about the name and accession, it's important to know where a cell line comes from. So we want to look at where it comes from in terms of species. If it's not a human cell line, the breed and subspecies, that's important. For human cell lines, the population, we'll speak about that because there are ethical issues about it. Also genome ancestry, HLA typing information, but of course, and that's quite important, the sex of the donor and the age at sampling, and the anatomical origin, tissue or an organ. Let's see all of those different things one after the other. Species of origin is quite easy. It can be indicated using the NCBI tax IDs. The NCBI taxonomy database is quite comprehensive. We only had to ask once for them to create a new species entry because we had a cell line from a species which was not yet in the taxonomic database. So in general, there's no issues there, and we can use the NCBI tax ID. One thing you should know is hybrid cell line and hybridomas, by definition, can have more than one species of origin because they're produced by the fusion of two cell lines, which can be from different species. And so, in fact, if it's from different species, you can have more than one species origin for that cell line. And we have about 900 entries which come from 2 species and about 10 entries from 3 different species. Now, this information is present in all entries. By definition, you have to have, this information. So there's a few cell lines where we don't know which species they come from, but fortunately, there is an NCBI tax ID for an unidentified species. So there's no problem annotating this. Okay, species, no issue. Now, for animals, you can use breed and subspecies. You can add breed and subspecies. Unfortunately, it has to be free text because there is no global database of breed and subspecies. Also, unfortunately, for animal cell line, it's often difficult to find this information as it's not, listed in papers. Quite rarely do you see this information in papers. Often you have to go back and ask the authors about it. We have about 750 different values for 18,300 different animal cell lines. Here are some examples. And those examples come from different species. You recognize a mouse strain line, strain name, a breed of dog, a crossing of 2 breeds of beef, a zebrafish strain, and a Drosophila strain. Drosophila, strain. Now, population information. This is for human cell line, and it's a tricky subject. We originally didn't and thought have this information in Cellosaurus and thought it should not be included because it's an ethical minefield to use term applied to a population, not to speak of ethnicity or race. But then we saw in the last 2 years that there is a lot of reports that have emphasized that the lack of diversity in genomic data set, which is highly skewed toward individual of European descent, is hampering progress in precision medicine for non-Europeans. This is an article in New Scientist a month ago. And in fact, it becomes important to note if the cell line comes from a non-Caucasian population, because it can be useful for people doing research, medical research, for those non-Caucasian population. So we started entering this information, and we have a small, evolving set of terms describing population. Here you see some examples. And only 30% of human cell line entry are associated with population field. So it's a work in progress. We'll never get to 100% because, of course, a lot of cell lines, this information is not available. But you have to know it's a work in progress. Genomic ancestry, I will go quickly to it because it's, a set which is limited in terms of scope, but it's a study which was done by Julie Dutil Group in US, which has a site called ECLA, Estimated Cell Line Ancestry. And for about 1,400 cancer cell line, they have computed the estimated ancestry of the donor, comparing those exomes to the genomic information from the 1,000 Genome Project. And they, consider 7 different ancestral populations. And here is the example for ELA, where you see the mixture for the ELA exome of 64.7% African, 0.7 Native American. I'm not going to describe this. You have to look at the paper and look at all those computations are done, but it gives you information based on genomic sequence of the origin, genomic ancestry of the individual, which gave rise to that cell line. Now, one thing which is also useful is HLA typing. Again, I'm not going to go in detail. Of course, those slides, I didn't say at the beginning, but those slides will be also, linked to the documents that, Google documents, and you will be able to have those slides, so you can go back to it. And we have HLA typing information for about 3,000 cell line. Here is an example of how it looks like, which you have MHC class 1 loci, MHC class 2 loci, and so on. And of course, the degree of precision of this typing depends on when the typing has been done. Some typing has been done a long time ago with unprecise data, or if it's been done using sequencing data more recently, and then it's quite precise, and it uses allele information, which can be very, very precise. So is a question? No, okay. Now, sex of donor, that's very important. And we use the control vocabulary, which is composed of 5 terms: female, male, mixed sex, sex ambiguous, or sex unspecified. Now, of course, everyone understands what's female or male. Mixed sex is used for animal cells less than 50, where the donors was a mixture of animals of different sex. Believe it or not, people have created some cell line where they've used, for example, a litter of mink, for example, and just homogenize all of those mink embryos and created cell lines which sometimes are a mixture of different sex. Sex ambiguous is used for cell lines that originate from individuals with various forms of congenital anomalies of the reproductive and sexual system. And of course, sex unspecified means that we didn't find the information, but it can be later updated if we find new information. And we don't annotate hybrid cell line and hybridomas with this type of information. Now we come to 2 different types of information items, which are still important to know where cell lines come from. So age of sampling, which can be either a precise value, 5 days, 2 weeks, 6 months, 1 year and 3 months, 15 years, 3 fetal months, or a developmental stage. blastocyte stage, fetus, children, adult, or for non-human species, you see here an example, lamb, fry for fish, 15-star larval stage, and so on. And when we cannot get this information, it's annotated as age unspecified. Again, hybrid cell line and hybridoma are not annotated with this information. About 85% of the entry contains this information. And the oldest donor, this is just anecdotal, the oldest donor for human cell line was a 184-year-old woman. Now, you will see later when we speak about future developments that we want to tie this information to developmental ontology, developmental stage ontology. So we'll get back to this later. Last thing is, and which is quite important, is an anatomical origin of a cell line. Which tissue, organ, or cell type does it come from? We have two common fields which describes this, called derived from sampling site and from metastatic cell line derived from metastatic sites. Here is some examples. And this is a work in progress. Again, this data did not exist until about a year ago. And only 2/3 of the entry have been retrofitted with this information. And the next step is to link the textual description to the Huberon anatomical ontology, which is cross-species. Okay, now we go to something very, very important, cell line integrity. And we'll see the issue of cell line contamination and misclassification. We speak of ICLAC, annotation of problematic cell line, what are STR profile, and what do we do with those STR profile in Cellosaurus? Now, you may have heard of cell line contamination because people have spoken about it quite a lot in the recent years. Those are just titles of articles. And the estimate is that, for example, for cancer cell line, contamination could range from 20 to 36% of all cancer cell line which are used. Now, here I need to backtrack and explain and do a little bit of nomenclature here. Now, what do we mean? Some definition are needed here. Contaminated cell line arise from two different types of process. Either cell line can be contaminated by another cell line, a foreign cell line. So this is what people generally call cross-contamination, or they can be contaminated with microorganisms, microbial contamination, generally by mycoplasma or fungi. Now, the second one, I won't speak about it because this is something which happened quite a lot, but people can generally deal with it. They can start back from an old batch. They can cure the cell line of those contamination. It's a well-known and huge problem in terms of growing cell line, but it's not something which is important to discuss in terms of a cell line database. I mean, a cell line can be infected by mycoplasma, but then, basically it can be cured of this infection, or you can go back and restart your culture without this contamination. So big problem is when you have foreign cell line contamination. And then you have misidentified cell line, which are the result of error in the species or gender. People have thought a cell line came from human, but it was not human, or they thought it was from one species and it comes from another one, or they thought it was a male cell line, but it's a female one. And then there are misclassified cell line, where the tissue type or cell type or disease is incorrect. Here are, well, first here is a cartoon in a website of a company showing, that when people generally find out that their cell lines are contaminated, it can be a lot of grief. And here are three screenshots of titles of recent paper from last 10 years. One is only 3 months or 4 months old, which shows the 3 different type of problems. Here a cell line, which was thought to be a urinary bladder cell line, but unfortunately it's HeLa. Here, very recent, they sequenced the mitochondrial genome of a cell line, which was thought to come from one monkey species, Sarcopithecus aethiops, but it's not from that monkey, it's from another monkey. And here, third example is a cell line, which is from human. It's a cancer cell line, but it's not what it was thought to be, which was a seminomona cell line. It's totally different. So what can we do with this? Well, first, you have to know that there is an institution, well, a committee called ICLAC, which is the international, sorry, I forgot the international here, Cell Line Authentication Committee. So the word international is missing here, which was founded to make cell line misidentification more visible and to promote awareness and authentication testing as effective ways to combat it. So what does ICLAC does? It curates a register of misidentified cell line, all kinds of cell lines which have problems, the three types of problems I mentioned before. But before being included, it needs to be carefully appraised by the committee. So each cell line is seen by the committee members, which look at the information which is available to them. Sometimes they try to get more information, either from people which have used a cell line, established a cell line, or from cell collection. And once, things have more or less, converge into an answer, the committee votes to enter the cell line into the table of misidentified cell lines. So it's two tables, I won't go in detail. One are cell lines which are misidentified and there is no real stock, original stock of the cell line left. And some where some stocks are contaminated, but the original stock still exists. So, but I'm not going to go into those details right now. And currently a lot of those, I could say the majority of new candidates are identified through the process of creating cell line information Cellosaurus, because we just happen to find or being aware of a lot of those issues. You can go to ICLAC, look at the different data sets and look at the register of misidentified cell line. Now in the Cellosaurus, what we do, we use a field called problematic cell line, which I show here three different examples, one for a contaminated cell line, an ELA derivative, one from misidentified cell line, a cell which was thought to be human, but is from pig, and one from misclassified cell line, which was thought to originate from a neuroblastoma, but which is from an Ewing sarcoma. So you have this information about the fact that it's contaminated, and we also put this type of comments in the descendant of the problematic cell line. If cell line have been established from a problematic cell line, then we will say the parent cell line, the grandparent cell line, the ancestral cell line has been shown to be XYZ. Now, if the problematic cell line is part of the ICLAC registry, you will have a registration comment which points to the cell line authentication committee register with the accession number in the committee line. Now, there's about 1,000 cell lines which are notated as problematic. So you see quite a lot of them. And of course, you can also browse them directly from the main menu of Cellosaurus, where you have this option, browse problematic cell line. You click on it and you have the list of those contaminated cell lines. Well, I should say problematic cell line, because as I said, it's contaminated, misidentified, or misclassified. Now, that leads me to the STR profile. So what are those? Those are short tandem repeats which exist, in the human genome. As you will see, they exist also in other species, but they were first well-defined inside the human genome. And because they're polymorphic, they're quite short repeats and quite polymorphic, they're used in forensic identification or for paternity testing. So all those movies or series where you see people trying to identify someone using DNA are based on STR, profiling. But they can also be used for quality and integrity of human cell line. So there was a standard 10 years ago, which was published, which proposed using 8+1 loci. I will explain why 8+1, but so 9 different parts of the genome, which can be used to, identify cell line. This standard is being revised, and the draft goes in direction of having 13+1 loci. Many commercial kits will allow you to test for 18 different loci. Here you see the list of those loci, and why I say 8+1 or 13+1 or 18+1, there is one loci which is just for gender determination. you have either the X version of amelogenin or the Y, X plus Y version if you're male. And so it tests for X or XY using this loci. The other one are real, short tandem repeats, and they basically have different allele values. So what do you do? Well, yes, I forgot also to say, this is for human, but very recently, STR markers have been established for mouse and dog cell line. No other species yet, but mouse and dog, yes. So what do you do? Well, you take your cell line and you send it to either your biomedical research facility or to a company, and they will run the STR profiling, looking at the different allele for the different loci, like here, D21S11 has a peak at 31.2, another peak at 32.2. For D18S51, you have a peak here at 15 and 16, and so on. And they will tell you if your cell line is what you think it is. The problem is most of those company or cell line collection which do this keeps a database of STR profile private. So until the Cellosaurus started getting this information in it, you had to go to a lot of different places, go to ATCC, go to the SMZ, go to ACSC, and try to see and run the STR profiling tools or ask for your cell line to be identified. Now, what we did recently, well, recently, no, 5 years ago, is we started capturing STR profile information for a lot of different variety of source, cell line collection, publication, and more direct submission. So most of those cell line with STR profile are from human, because those are the biggest set of cell line where people are doing STR profiling because of all the problem with cancer cell line. Only a few of them are from mouse and from dog. You see only 74 for mouse and 36. But this will increase quite quickly for mouse because it's only 1.5 years ago that the STR profile were defined, the loci were defined for mouse, and it's only a few months that labs are testing for those and that companies are providing now mouse STR profiling. So this will increase. And of course, we indicate for a given cell line all the different sources that contributed to build the profile. For example, here is a cell line which has information coming from 6, 7, 8, 9, 11 different sources. And of course, there are conflicts between those sources, which can be due to errors, but more frequently to genetic drift of the cell line. A cell line can lose or gain an allele, and so it's important to note this. Here is an example of an STR profile, and you can see that here the cell line seems to be from male. It has X and Y, but two cell line collection have not found the Y chromosome in this cell line. The amelogenin Y, loci. That happens quite often. It's easy for a cell line to lose the Y chromosome and thus amelogenin Y loci. Or here, you see that 4 resource find 10, 13 for D5F18, but 1 resource find an additional loci, which may have been lost or gained by in the cell collection, which reports it. So that's quite important. So cell line STR profile.
Amos, there is a question too in the chat is regarding ICLA. ICLA. Yeah, ICLA. Currently, 552 cell lines. Are there additional ones on Cellosaurus?
Yeah, there's 2 reasons. First, because as I said, we annotate descendant of cell line which are, which are problematic. And in ICLAC, you will find this only with a few cell lines. They have, I think, 8 or 9 entries which are descendant of other cell line, but they don't systematically report all of the cell line which are descendant of others. So that adds probably about 200 to the 500 or 300 to the those which are, in ICLAC. The rest are cell lines which have been now seen by ICLAC. Those are things which are, it's an ongoing process, as I said, and it's enters the Cellosaurus and at the same time goes to ICLAC, but it can take time to get an ACLAC registry number, because as I said, careful work has to be done to really double check everything. And so in fact, but the main discrepancy is that one is the fact that we annotate descendant of a cell line which is contaminated.
Mm-hmm. And there's a comment from Janet saying for the STR cell line typing service checkout. So there is a website, microsyn.ch.
Yes, that's one of the company which does it. It's a company in Switzerland which does it. A lot of companies do that. I mean, I didn't mention companies because I don't want to advertise one specific company over another one. I mean, I know this company very well. They do a lot of good work. So I don't have anything against them putting this in the chat, but it's only one of many companies doing a very good work of STR profiling. I mean, there is a lot of players in STR profiling, but this company is really doing good work. So it doesn't matter if they advertise themselves in the chat.
Thank you.
So cell line properties, that's more wide subject where we will see 6 things, which are listed here: category, characteristic, biotechnology, doubling time, karyotypic information, and microsatellite instability. Category, it's easy. We basically, every cell line is assigned to one of 14 defined categories. You have here's a list of those categories that we defined, and what percentage of Cellosaurus entries belong to this category. So you see transformed cell line and cancer cell line are the highest, followed by embryonic stem cell, induced pluripotent, and so on. I mean, the first 6 or 7 one are the most, frequent one. The rest are more, anecdotal. As I said here, transformed cell line is by far the largest category, mainly because of the ease of creating EBV immortalized B lymphocytes. So people can very quickly take a cell line from a peripheral blood of an individual, use Epstein-Barr virus to create an immortalized cell line, and that works quite well. And so this is used a lot for genetic disease cell line, where it's easy to do this. But you see cancer cell lines come second, and induced pluripotent stem cell are growing quite rapidly because people are developing more and more iPSCs for a lot of different applications. So that's easy.
Question, sorry. How is about the STR. How STR profiles being performed in mouse cell line? Is there a commercial kit for that?
Yeah, there is. And in fact, Microsynth, which was just mentioned, does that. There is, if you can buy a commercial kit, this I don't know. There are companies which are doing STR profiling of mouse. There is a paper describing this, in details. We can put this later inside the Google Doc, so paper on the mouse and dog cell line papers. But I don't know if there is a kit yet, a commercial kit or not. Okay. But there are sure companies doing it, including Microsynth. Now, characteristic and biotechnology, at the beginning, there was no textual information describing the cell line in Cellosaurus, and we started having small blurb of text which describes the characteristic of a cell line, like here, useful for the study of drug delivery and phagocytosis of pathogen and so on, or grow stably at 35 degrees Celsius. Or here is from a biotechnological use of a cell line, an example for MRC5, which is used for production of different vaccine and so on. So this is text, and it's something which is being added, more and more into the Cellosaurus. When I create now a new entry, I like to add this type of information, or when a cell line is retrofitted with information, because it gives a little bit an abstract about the characteristics of a cell line for people wanting to know more about it. Just text. Doubling time is important. So we capture the population doubling time of a cell line, and it originates from either publication or cell line collection. So it's generally, in hours or days. Sometimes here is just example with hours, but it could be a number of days also. Should give an example with days instead of hours. So we'll beef up this example. and about 6,500 cell line contain such information. Again, this information is not seen everywhere. I mean, DSMZ and ATCC give it, DSMZ is quite good about this. For every cell line, they have 800. So it gives this information, ATCC only for a few of them. And sometimes you find them in publications, sometimes not. Karyotypic information, this is something recent. This is not meant to be something to capture a precise textual description of a cell line karyotype, but just some noteworthy features. So I'm not going to go in details there because it's more anecdotal. It's to generally describe something like almost interesting in terms of the karyotype, like if a cell line has lost chromosome Y, or like here, this is created from a patient, which is mosaic for 2 types of cells, and there is a cell line for both type of cells for this mosaic patient. Or a cell line which has expanded in the number of chromosome compared to its parental cell line.
There's a question also from, do you provide a translation from Brenda tissue ontology to Cellosaurus and back? And is there an ontology for Cellosaurus?
So the first thing is that if there is cross-reference to Cellosaurus.
Yeah, I guess it's a cross-reference from Brenda tissue ontology to Cellosaurus and back.
There is cross-reference to all the cell line ontology. We'll see that in a few minutes.
Okay.
But since there is no reverse links, I mean, Brenda is not putting links back to the Cellosaurus. No cell line ontology is putting, for a moment, I mean, cross-reference back to Cellosaurus that I know. A lot of database of cell line are putting links back. Cell line collection are putting links back, but cell line ontology do not do it. Mostly because they probably don't really put cross-reference to database. They don't have really cross-reference to any database. So microsatellite instability, I mean, I put here the definition. It's a predisposition to mutation which result from impaired DNA mismatch repair. And so this is quite common in colorectal cancer cell line. And there's a classification between MSI high, macrostability high and low. And so you have this information which is captured inside the Cellosaurus, but only for 1,400 entries because, I mean, most cell line are not tested for that. And it's only cancer cell line which are tested and mostly colorectal cancer cell line. Now, one thing which is important is cell line are engineered quite often because they can be transformed from the original cell, transfected or transduced. They can be knocked out. They can be selected for resistance. And there are other things which can be done to them. Now, I'm going to go quite fast into that because I'm running out of time. Well, I still have 5 minutes for this section, but I want, what time did I say I wanted to pause? At 4:20, no?
Yeah, 4:20.
To 4:40, I said, okay.
Yeah.
So I'm going to go a little bit faster here. So there is a variety of ways you can immortalize cell line using physical means, radiation, chemical, and viral. And we describe this with a structured, field, and where there is a database, an accession, and description. Here is some example. One example of a chemically transformed cell line with a carcinogen. One example of EBV transformation, which I discussed before, or one example of radiation transformed field. And this is used both for artificially transformed cell line and for cancer cell lines that have arisen through viral carcinogenesis. So you can transform a cell line, but of course you can transfect or transduce a cell line by introducing foreign gene. So it's called transfection when you're using a chemical carrier or transduction when using a viral vectors. So this we also annotate with a structure comment with which gene was transfected or transduced inside the cell line. Now, one thing we do, we're using currently transfected with for both transfection and transduction. So a more generic term would be more appropriate. And if you have suggestion, I'd be interested because I couldn't find a good term which can replace both transfection and transduction, except a sentence like introduction of a foreign gene, which is not really nice. So if you have a word to replace transfection and transduction to one term which describe both, I'd be willing to use it immediately. Knockout, of course, instead of putting a new gene, you can also knock out a gene. And again, we use a structured comment with a method, a database, and an accession description. And of course, popular co-methods, there's a lot of different methods, but one of the most popular one recently is CRISPR-Cas9, but it's not the only one. And here you see example of knockout lines, CRISPR-Cas9, CO, or TALEN. And you can also select for resistance to, drugs, and that's useful in cancer biology. People are trying to develop drug-resistant cell line. And again, this is structured where we say which is the database of the chemical and the accession or description. Here you have a description of a cisplatin resistance or cetuximab, which is a monoclonal antibody. Here's a third example is for something which is not for cancer cell line, but you can also, establish toxin-resistant cell line, like cell line resistance to the ricinus toxin or to diphtheria toxin and so on, or to radiation also, resistance against, radiation damage. So we have about 13,000 cell lines which are resistant to a compound or to physical process, about 226 different compounds. Just two last things about it. When we describe disease in the next, I think it's the next thing, we'll basically speak about the sequence variation field. And we use also sometime information about how a variation has been introduced using a method like here, CRISPR-Cas9, or when a mutation has been corrected using, a method, engineering. This should be modified to be, sorry, this should be not, this is not optimal and should be modified to be, putting not the characteristic comments, but sequence variation comments. Okay, quickly, cell line groups and panels. Very quickly, because that's easy to understand. We put a tag which groups together cell lines so they can be retrieved easily without the need of performing complex queries. So we created groups for a series of taxonomic range, which people like fish, insect, non-human primate, amphibian, and so on, and some intended use of a cell line, like hybridema fusion partner, vaccine production cell line, and so on. And we have 5 other groups which are not specific to one of those 2 categories here. Last one is interesting, endangered species and breed, because more and more cell lines are going to be developed to be used for conservation of organisms by using pluripotent stem cells to keep the germ, form of endangered species to rescue them or to preserve them. Here is an example with a cell line which is both a non-human primate cell line and which is from an endangered species breed and so on. It was established from a monkey which was trained to accept regular blood sampling and insulin injection. And of course, you can query those directly from the Cellosaurus on XPASI. And the same principle applies to panel, where we have a command called part of, which can be used to select all the cell lines which belong to a panel. The most well-known panel is this famous NCI 60 panel, which had 60 cell lines, 58 really, because 2 were contaminated, which was used for the last 30 years to test anti-cancer compounds. But we have identified more than 100 panels. And again, you can query them from the Cellosaurus by saying grows by cell line panel. Here just a short part of the panels, just the first 20 or 30 panels you see by alphabetical order. It goes only to E, A to E. Okay, disease information. That's quite important. So annotating disease with, we annotate disease, as you will see, with NCI thesaurus and orphanet ordo. So there is what is called an NCI Cellosaurus value set, and we annotate sequence variation. So first thing is annotating disease. We use two medical ontology to annotate the disease affecting the donor of a cell line. NCI thesaurus for all disease, human and non-human, and orphanet ordo for all human rare disease. So you may wonder why two. So NCI thesaurus covers all of the disease, and we have a collaboration with them. When we needed disease terms that doesn't exist, they created, and they created about 1,000 terms for, or not 1,000, 600 terms now for us. So you could say we don't need another ontology, but the rare disease community has asked us to also use orphanet ordo for all human rare disease. So we have also added recently the information on with orphanet on disease with orphanet ordo ontology. And about 58%, so it's a lot of cell line concern, I mean disease, I mean either human disease or animal disease, because 58% of all Cellosaurus entries have at least one disease term. And we use about 2,000 different NCI terms and about 1,150 ordo terms. So three things you should know. We do not annotate cell line arising from carriers of a genetic disease with disease term. So if someone like a parent of a diseased child is a carrier of a mutation but is not sick himself or herself, we do not annotate them as with a disease term. We may change this and have a system of annotating disease which will be more complex and which will have this information. Cell line originating from non-cancerous tissue in a cancer patient are not annotated with disease terms. That's logical because they're not cancer cell lines.
There's a question about the disease ontology. What about disease ontology, or why not use the disease ontology?
Because it does, that's a very simple answer, because we needed disease which are not only human. Problem with a lot of bioinformatic resources that's specific to human. So disease ontology is perfect for human, but we couldn't find about 500 terms which deals with dog cancers, rat, lymphomas, zebrafish disease, and so on. You have to think that the Cellosaurus is not human specific. It caters for every species, and fortunately, NCI thesaurus has information on non-human disease, and they accepted to enter new terms. So that's why we had 600 new terms in the NCI thesaurus, because we entered all those terms and they could do them very quickly. Once those terms exist in the disease ontology, we can also link to the disease ontology. So also cell line edited to correct disease-causing mutation are annotated with disease terms. Even so, you could say that by correcting some mutation, you correct the disease. But it's a philosophical point to know if you have a cell line which has a disease mutation and you correct it, should that cell line also have this, cross-reference to the disease ontology? Again, this should be modified in future. As I said, NCI has created what they call value sets, and they have one they call the Cellosaurus disease terminology, which allows you to get all of the terms which are used in Cellosaurus. Now, sequence variation is quite important. So we try to capture relevant sequence variation about genetic disease causing mutation and important oncogenic somatic mutation. Important is important here. By this, I mean we don't want to recreate cosmic. So we're not going to capture all somatic mutation in cancer cell line. People can go from the Cellosaurus to cosmic to get them. We also want to have gene deletion and amplification and gene fusion. Again, it's an important one. So we describe them using the HGVS nomenclature. When a variation exists in ClinVar, about 50% of the variant we describe will link back to relevant ClinVar entry. Here I have an example of an entry which has gene fusion and a number of mutation, either homozygous or heterozygous, with some of them being in ClinVar. I didn't say it, but we also have linked to dbSNP if it's not in ClinVar, but there's not so many which are in dbSNP and not in ClinVar. And then what's important also is okay, 12% of all entries contain at least one sequence variation. We now have 6,000 different variations that affect more than 1,000 genes, 95 in human cell lines. Again, it's not only human specific, we have mutation in non-human cell lines. And it's a work of progress. So we want to still continue formalizing them. And we have not yet caught up with retrophitic cancer cell line with oncogenic mutations. So it's not, a final way of representing this information. We're going to change the way the zygosity of the cell lines, the gene here should be tagged with the AGNC accessions. The zygosity should be using a zygosity statement and so on. So this is all going to change in the near future. Now, here I'm going to skip this because monoclonal antibody-specific information is only for antibodies for hybridomas, and most of you are probably not working on it. So you can look back at those slides later, or we have information about the monoclonal antibody isotype and the target, which protein or which chemical compound is a target of that monoclonal antibody, which is produced by an hybridoma. And there's about 5,000 hybridoma with this target information. Now, I got here 7 miscellaneous fields, which I'm not going to go again in a lot of details because of lack of time. I'm going to concentrate only in 3 of them, discontinuation and caution and entry story. So registration is useful to indicate that a cell line has been entered in an official registry. I told you about the ICLAC register or registry, but there is other registers that exist for embryonic stem cell or for a patent. When a cell line is described in a patent, so people writing the patent, the company or the individual needs to deposit their cell line into an international depository authority. And so this is registered then in this the IDA, which is often a cell line collection also. But here you see the Pasteur Institute collection, has registered a cell line with this number I4403. Discontinuation is important because when a cell line is no longer distributed by cell line, we indicate this, and there's only one to do it. On the XPASI site, it's merged with the cross-reference to the cell line collection. So of course you cannot click anymore because the cell line doesn't exist anymore in that collection. But the problem is almost no data collection, cell line collection track publicly discontinued cell lines. So we're de facto acting as a repository for such information. And we have about 9,000 discontinuation information statements. Anecdote will skip. We'll skip caution. You can read about it later. And we go to also the omics field will be skipped. And we go to this important field, which is entry story. Each Cellosaurus entry contains a record of when the entry was created, when it was last updated, and number of versions it went through. Like here, this entry was created in 2012, updated in 29 of October 2020, and 33 versions of the entry. Here is a graph showing you that, the majority of cell lines, here 20,019, 17,000, 17,000 have gone through 3, 4, 5, 6, or 7 versions. So you have very few cell lines which have been updated at every release. It's normal. Only a few of them, 29, have been updated 37 times because there's 37 release, but a lot of them have been updated 5 or 6 times. This is important, child-parent relationships or sister autologous cell lines. So that's 2 relationships we do. Cell lines which are derived from another cell line have a hierarchy or derived from in Zobo format statements. So for example, this cell line, which is derived from MCF7 as a hierarchy derived from statement. And of course, hybrid cell line can have more than one parent. And about 30% of all Cellosaurus entries describe cell lines which are derived from another one. Here is an example of all the children of EPG2, which themselves can have children, which themselves can have other children. I mean, sometimes it can make an aesthetically pleasing image. Here you have MCF7 and a lot of cell line which is derived from MCF7, one of them which himself was used to derive other cell line and so on. So you can create like this interesting image of the parent-child relationship of cell line. Now, sister or autologous cell line are derived from same individual. So each cell line which is derived from same individuals than others will have as many originates from same individual statement as it has sisters or autologous cell line. Like this cell line will have two statements because those three cell lines are from the same donor. And of course, in the other cell line, you will have statement going back to the two others and so on. It's, I mean, of course, I mean, each has the information of which are the sisters of that cell lines. 10% of the cellular entries describe cell lines that have one or more sister cell lines. Now, this was a question before. Do we link back to those resources? Yes, of course. We have cross-reference and web links. Cross-reference, we link back to a huge number of resources. Exactly how many? It's 93 different resources which we link back. And we have 382,000 cross-reference, so an average of 3 cross-reference per entry. And we cross-reference to either specific cell line, resource like cell line collection, cell line database, or ontology. Okay, one of it is specific to cell line, but the other one here is not. So here you have not a list of those 93, but just an explanation, examples of different category of cross-reference, cell line collection, database, ontology, sample resource, proteomics, and so on. And when you're on the website, you have those categories shown, like here, you have cell line collection links, you have resource, ontology, BTO, CLO, EFO, biological resource, chemistry resource, and so on, polymorphism, cosmic here, and so on. So web links is another way to have links outside of the cells. So why is it not a cross-reference? Web links are just URLs. And the difference between web links and cross-reference is they go to cell line resource that have either very few entries, so we didn't want to create a cross-reference, or have inconsistent URLs. A lot of cell collection have URLs which are not based on a stable identifier, so we cannot create a cross-reference. And some goes to generic page describing research project using cell line or group of cell line. Last thing, and I will finish that part now in 2 minutes, is reference. So we want to reference where the data comes from. So where do those papers comes? We don't want to capture every paper which make use of a cell line. This would be completely crazy. I mean, you would have probably 100,000 paper just for ELA. So the idea is to capture the paper which describes establishment and characterization of a cell line. So that's where you have information which is interesting on that cell line, not every paper and not a paper which just use it without doing anything with it more. We cite about 21,000 different publications. All papers cited have been curated. And here are 2 types of statistics. First, we distinguish 4 types of publication. Journal articles, which is a majority, 93, or not only majority, it's it's basically a great majority of publication are journal articles. Book chapter or books or meeting abstract books, which is only 1%. Thesis, not only PhD, but BSc, MSc, veterinarian medical thesis or medical thesis, and patents, which is the second largest set of publication. And if you look at it, a reference can have 4 types of identifiers. That's important in terms of bioinformatics. You can have a PubMed ID, a DOI, a patent number, And then for all those which are patent and don't have a patent number, we assign ourselves an identifier called CLPub with a number. Those are only for those which we could not find a unique identifier. And you can see some statistics. About 80% of all the reference have both PMID and DOI. Some only have PMID only. A few percentage have DOI only. So those are papers on fish and insect cell line, because PubMed and Medline before was not abstracting papers which deal with fish and insect biology. Now they do so, but a long time ago, they were not doing so. So there's a huge amount of literature for fish and insect, which is not in PubMed and Medline. And only 2.47% are those CLPub. Here is a growth, and that will be the last line of this part, of the number of references cited according to the year of publication. You can see that it grew until the late 1980s, and it plateaued, went down and went up again. Why is that so? But well, now it's increasing again because a lot of papers are reporting new iPSC cell lines. And so it's increasing. And one of the reasons it decreased here is probably also an annotation lag that I didn't catch up with all of the papers from this period and so on. But basically there is a new peak of papers due to the fact that people are describing a lot of new iPSCs, and there's one journal, Stem Cell Research, which is publishing about 50 or 60 papers every month, which are quite short papers, each of them specific to one or two cell lines and so on. So let's start with the fact that the Cellosaurus is available on XPASI. Here I can jump to, and most of you know it because anyway, if you Google, you will find it. And you have the homepage. We will change a bit the homepage in a few months, but not drastically, because for the moment we don't have really the resource to make it much nicer. And the idea, on the homepage, you have things that we already saw, like you can browse by cell line groups, you can look at the problematic cell lines and so on. But what most people will do, of course, when they are on this page, is search for something, and often it's a cell line. I will go back to the search option, but here what I will do first is, once you have searched, what do people do? They generally browse for a cell. And here I've specifically took one example of a cell line which is contaminated. It's an ELA derivative. And you can see the different type of information we spoke about, the name, the synonym, the accession, which is the same as the resource identification initiative error ID. All of those different comments, some of them we spoke about, some we didn't have really, but here you have population information and so on. You have the disease, the species, the parent and children, the fact it's a female cell line. As it's from ELA, it's from a 30-year-old and 6-month-old individual at sampling. Henrietta Lacks, it's a cancer cell line. You have the STR profile, those web links, those reference. I'm not going to go to this list of reference, just go to it, and then the cross-reference. Now, there's nothing special to say there. Anybody can browse a page, and so that's straightforward, and people can look at that page, and generally, they don't have really question about it because information is straightforward and labeled, and we generally do not have any user questions about those pages. Yes, they can be made maybe nicer, we'll see about it, but for the moment, I would say they do what they aim to do, which is to show the information you have for an entry. So if I go back to my slide, yes, so what's the homepage here? I told you already it's available since already mid-2015 on XPASI. So it's about now almost 2,500 up to 3,000 sessions per day. Already 7 million page view, 2.4 million sessions. So it's used quite a lot on the XPASI server. And here I had screenshot in case it didn't work of this exact same line I show you. So I will skip those screenshots because we just did this. Which lead me to text search. Here I search for, a name of a cell line. It's obvious, it's easy, but you may want to search for things more involved than just the name of a cell line. And we have currently I mean, a full text search. So search is built on an index, which was built using Apache Lucene, which is a type of Lucene type search engine for those which know what Lucene is. And of course, you can search for a cell line name, but you can search for any other things which is in the text of the records of Cellosaurus, like the catalog number in the cell line collection, the disease name, the species, the reference authors, the titles, PubMed ID, sequence variation, transfected gene, and so on. And this, where you can search, is available on top of every page. You saw it before. If I go back to the top of the page, here, I can search and do whatever search I want on that part. And I could, for example, put an accession number of a cell line or whatever I want, a PubMed ID or everything else which I want to put.
There's a question.
Yeah.
On how much attention do you pay for vaccine production cell line updates? When I search that in Explosive right now, I find 89 results. Can I consider this number as very actual for January 21? Thank you for your answer.
Yes and no. Yes, in terms of those where the vaccine is commercially produced. No, for in term, well, yes, for two things. For those where there is a virus, a vaccine produced. So if the vaccine says this cell line has been used and so on. And yes, for those where cell line has been, the case where cell line has been specifically developed for vaccine production, and it has been published as such, as a good cell line for vaccine production because there are papers, but no for all the cell lines which are being used to develop virus, but vaccine, but the vaccine are not yet available and nothing has been published about it. So, I mean, there's probably a huge number of derivative of some Vero or MDCK or ACT293 or other cell lines which are used, but they are not captured because information is not available.
Thank you.
So to go back to the search, one thing you should do is use, and that's maybe obvious to say, you should use precise terms to search in Cellosaurus. So the best thing is if you're searching for something which is linked to an ontology or control vocabulary, you should use that term instead of the text version. For example, if I want to look for dog seg line, I should look for 95615, which is, so I can say tax ID 9615 instead of, just 9615, and have, cell line from dog instead of saying just dog, because I will get anything which has inside the title of publication or elsewhere the word dog, like this mouse cell line is not from dog, but I guess that if I go to the record, I will see the word dog somewhere on it. Okay, that's obvious. You need, for example, another, maybe not so obvious, you want to look for SV40 transferred cell line. If I do SV40, I will have a number of cell lines, 2,767. Probably most of them are SV40 transform, but if I look at the tax ID of 1891767, sorry, I cannot cut and paste here. I should get here, I get less, 2,332 instead of 2,000, what was it, 2,700. So 400 less cell lines, but those I'm sure are SV40, transform, because they will have the tax ID of the simian virus 40. Okay, so that's obvious for most of you. And here I give two more examples. But I know by looking sometime at the log on Expasia of people searching that a lot of people are searching with very fuzzy terms. And of course, they don't really get what they want. They get more or less depending on what they're searching for. Here is three examples where I won't do it now on the web, but you can just look at those things. It was meant as example for people working on the rare disease, that you should not use the name of the disease to search for. If I search for cystic fibrosis, I get 302 hits, but 20% of those cell lines are not from patients suffering from cystic fibrosis. So the word cystic fibrosis may have been in the title of one of the paper or elsewhere. And if I do the search with Orphanet 586 from, classification of disease, then I get 239 cells, which are, I mean, specific to the disease. And of course, you can, as I said, look for, I mean, a sequence variation using the HGVS nomenclature. You could look for ClinVar accession, basically any type of information which is inside the full text. We will come back to that because, of course, this only allows a full text. It doesn't allow structure queries. And this is something we want to be able to do, but I will come back to it. But still, as a full text search, it works well if you use it in a nice way, basically by using it with the maximum number of precise identifier as possible. Now, one thing which is important is we have another search tool, which has nothing to do with the content in terms of textual content. But as I told you, we have STR profiles inside the Cellosaurus. And then the idea was to build a tool which allows to take your STR profile from your cell line or from anything you saw in a paper and run it against all those in Cellosaurus using a variety of algorithms which has been developed to do STR profile search. So Cluster was developed by Thibault Robin. I mean, he was a PhD student in my group. He developed this program which worked on human, mouse, and dog STR markers. It allows up to 31 different STR markers in human, which is all of those which are used by, I mean, by all different kits that exist. You have 3 different algorithms you can use, different computation mode. You can do this in batch mode. You don't need to do one by one. You can create a file with as many cells as you want and do it. And you have a RESTful API to do it also programmatically. And it's extremely fast. Here is the interface. What I will do now is just go back, do it directly on the web. And I go to Cluster. And I will take an example. Here it's just one cell line as an example. So you see the values of the allele are there. You search, by default it uses, I mean, one algorithm and one, I mean, and what do you call some, I can't find the word, some threshold here. And you have the list of results which you can export as a CSV file or as an Excel file. You have also a JSON file. Forgot that you could do JSON. You can see here best and worst. Maybe I need to explain this. And you can see things in red. Red is easy to explain. Those are a contaminated cell line. So here it shows you some contaminated cell line which have STR profile very similar to HT29 because they derive from HT29. But here you maybe don't see it, but it says best and worst for the same cell line. I told you that the STR profile information may have conflicts, variation depending on who did it. So the algorithm takes all the different permutation of the STR profile and compute what is the best match and the worst match that it can do with the data which is available. Quite nice. To our knowledge, it's the only tool which can do this, and you can skip, you can go to mouse. Sorry, I should go back to the, I'm not inside the right, I could go to the mouse tab, do this with mouse or with dog. And you can, I'm not going to go more in details of these tools, but I just basically managed to kick myself out of the presentation mode. Again, here you see the results, what I described. Now, one thing which I want to discuss also in 2-3 minutes, which is mundane to searching in Cellosaurus, is in 2017, we initiated a project to develop a bot, so a software tool that would populate Wikidata with cell line concepts. So each concept being equivalent to a Cellosaurus entry. So a student, Lelia Debond, developed this tool in 2018, and recently Tiago Ljubljana from Brazil has updated it. And what it does, it basically takes Cellosaurus data and tries to fit it to the semantic model of Wikidata. And basically there is in Wikidata as many cell line concepts as there is cell line in Cellosaurus. In fact, Tiago just emailed me a few hours ago telling me he's going to run it on release 37, I mean, which was released a few months ago, to run and update the data on Wikidata. And of course, not all Cellosaurus data could be modeled in Wikidata framework. and it contains currently the following type of information. Sorry. I'm not going to go in details because I'm sure that only a few % of the people, well, there's only 68 people here long gone, are users of Wikidata, but you see that it's only a part of the data. And it's a work in progress because maybe new type of data from Cellosaurus can be put into Wikidata if it's possible to model it in a way which is complete enough. For example, for disease, we needed to make sure that there was enough disease terms in Wikidata, which there is, which add an NCI thesaurus term or an ordo terms. And in a few cases, we had to add those terms so that we could link to the disease. Now, here is a Wikidata entry created with the Cellosaurus boat. And the important thing is, why do I speak about that? Okay, it's interesting for developers and people interested in Wikidata, but you can also use it thanks to the Wikidata search query service to do very precise search if the data in question is inside the Wikidata. So for example, you can do this type of search, which you cannot do currently with the full text search. I want all cell lines which are associated with disease that have an ordo identifier and that are children of the Wikidata concept carbohydrate metabolism disorder. So I'm looking for all cell lines which basically are rare disease and are part of the carbohydrate metabolism disorders. And in a few milliseconds, you get the results with the disease and cell line, and you can go back to Cellosaurus entries. So that's quite interesting. And it will lead to the future developments of having a semantic version of the Cellosaurus where you can do this internally from the Cellosaurus and not have to go to Wikidata. Now, you may want to download the Cellosaurus, and you're encouraged to download to use it internally. I mean, either because you have application which need it or you're writing something. I forgot to say it was in the slide where there was a Cellosaurus in one slide. The Cellosaurus is available into CC with a CC BY 40 license, so you can make use of it as you want. And you can download it by FTP. It's distributed in 3 formats: structured text, OBO, and XML. Some things need to be said about some of the formats. The OBO format file does not contain the steer profile data, the age at sampling, and the full reference records, because that would, I mean, we couldn't find a way in OBO to describe this. And all of the comments in an entry are concatenated because there can be only one comment field, again, to a knowledge inside the OBO format. So it means that there is huge comments fields where the structure is almost lost because it's a huge string of characters.
There's a question here, yeah. Sorry. From a license point of view, Cellosaurus is under CC BY 4.0 and Wikidata is CC0, if I'm not wrong.
Yeah.
Are there any issues between the two?
No, because in fact, also data which was, I mean, basically, there could be an issue if somebody writes a boat for a resource without asking permission to that resource. Yes, there is an issue because that resource could say, well, we don't agree that this data be put in a CC0, but being the one doing the Cellosaurus, we agreed to put this data in Wikidata, therefore there's no issue. If the one which writes a boat has a permission to do so, then there is no issue. So basically we accepted that all the data from the Cellosaurus, which was put into Wikidata by definition becomes CC0.
Okay, thanks.
But it's true that some people have taken resource and put them on Wikidata without asking permission from the resource and then put basically data which was CC BY or not even CC BY into CC0. But here there's no problem. So the structured text format requires 2 files, one with records and one with reference. And the URLs of the cross-reference are not inside the file. They can be built using the information which is in a 3rd file, which is called Cellosaurus xref, where the URLs rules are described. Now, the best way to use it is to use the XML version, because everything is in 2 files, and both the records of data and all of the reference records. And so Cellosaurus XML contains everything, including the instantiation also to URLs of also cross-reference. And it comes with, of course, an XML schema definition file. Of course, the cons of this format is that the file is huge because it contains all of this data, and XML is being quite verbose. There are some additional files which are available, but basically if you go to the FTP site, you have the list, you have a README, and all of those files, a list of all those files which are described here. I'm not going to go in details, but there are different files which can be useful. One of it is, of course, the release notes, which are also available from, sorry, if I go back here. The release notes are also found not only on the FTP, but you have directly the release information, which is here on the top page of XPASI. Okay, let's get back to, sorry, to FTP, XML. Now, one thing you may wonder is, yes, what you can do also, and I will show you this directly better, is if you're inside an entry and you don't want to download all of the Cellosaurus, but you want just to download one cell line or more than one, let's select this cell line, you just go to text version and you get the text version of the cell line. You have the record in Cellosaurus and the reference which are cited in the entry, which are there. And of course, this is with the REST API. So you see here that you can do this programmatically by using, I mean, so .txt for each accession number.
There's a question.
Yeah.
Are you considering also to provide directly a Cellosaurus endpoint allowing Sparkle queries?
Yeah, but that's going to be, yeah, in future developments.
Okay.
So here is what I said just before. You can have a URL in, I mean, in format with accession txt. So you can download, just clicking text version, and you obtain the entry and the reference record. Here is example again. Now, you can also get the Cellosaurus on GitHub, which all of the file except the XML is there, because the XML file is too big for GitHub. I mean, so, but you have since release 11, all of the different you can go back release by release on GitHub. And I mean, I would, if you're interested in old version, because you need to go back, I would go to Yareta, which is a research data repository of Geneva University and another higher education institution. And basically in Yareta, we created archive with every Cellosaurus release, starting with release 2. And so basically you have all of those releases which have been archived, except the OBO and XML file, which were not kept up to release 32. So, but you have the structured text file from release 2 to now, and since release 32, also OBO and XML. And you can download, so again, it's CC4, and there is a DOI for each of the archive. Okay, a lot of questions came to those, can be answered to those slides here, which is, what do we want to add? We would like to extend in terms of depth the Cellosaurus to cover patient-derived xenografts, because now it's only cell line, but you have xenografts which can be distributed, which could be useful to add. Now, another information which has been discussed is to add information on susceptibility of cell line to infection by virus. So this is not easy as it seems. We do have some type of information like this currently. If I go and go to any entry and take, for example, I will say, well, SARS-CoV-2, and I will have information here that susceptible to SARS coronavirus infection, susceptible to SARS-2 coronavirus infection. Okay, this is textual. It's not going to be parsed, and it doesn't give you information about how much of the virus you need to get the cell line infected. You don't know the growth kinetics. You don't know if it's cytotoxic and so on. So it's not, I mean, very easy to model what information should be captured for cell line infection by virus, but we want to do this in collaboration with the virologists and try to get a grant to do this. So this will be done only if we can manage to get a grant and have someone working on it. We want to add information on the biosafety level of distributed cell line. This is something that pharmaceutical companies have asked. And now let's go to something which is, and of course, I mean, now we go to something which is much more important, is to structure better some of the information. I told you when I was talking about disease, we want to formalize, continue to formalize the sequence variation information. We want to add links to Bairoch for tissue, organ origin of cell line for anatomical. We want to add links to developmental ontology for the age at sampling. And we want to restructure information on cell line availability. Now you have cross-reference to cell line collection. You have comments on discontinuation or the loss of a cell line and so on. And this should be done on something which is more structured. And now, this is where I was saying that a lot of questions went to here. We want to have an improved web representation of this Cellosaurus, forms for user to enter preliminary new cell line entries, and of course, a Sparkle endpoint to allow advanced search and federated queries, which means that we also, of course, we need to develop a semantic model, but we need also to have a total format distribution of the Cellosaurus. So we need to basically create an RDF version of the Cellosaurus, and at the same time have an API for querying and download of the resource. We also would like to have an alerting system for newly discovered contamination and misidentification, so that people can know which are the new cell lines which have been, well, they're not new in the Cellosaurus, but which have been newly indicated as being problematic. Okay, so I will end with 2 slides to thank people. Elizabeth Gastiger, which work in the web team of the SIB, has implemented the Cellosaurus on Expasis since 2015. Thibaut Robin was a PhD student which developed the class STR similarity search tool. Alain Gatto, which is the developer of my group, has developed a tool which is called Celloparser, which is both generating the OBO and XML version from the structured file, but also checks all of the syntax of the Cellosaurus to make sure that there is no error scripts in terms of syntax. So in terms of content, of course, these tools cannot deal with the content, but in terms of syntax. I already mentioned Lelia and Thiago for the Wikidata Cellosaurus boat. And there is somebody, I mean, Amanda Capes Davis, which her Twitter name is Cell Line Detective. She's the Secretary General of ICLAC, and she has had hundreds of useful suggestions over the year on how to improve the Cellosaurus. And of course, I'd like to thank all of the individual scientists, there's more than 500 now, that have answered my nagging emails to obtain additional information on the cell lines, and you for your attention. I just leave also that you can always send questions to this address, or you can go to the contact page on Expasis and click on contact. And you can follow the Twitter feed of the Cellosaurus.